Welcome to part 5 of documenting botanical diversity. So this is just something that came in handy that I thought was sort of a useful skill that I'd like to share. Going out in the field and collecting plants is a lot easier than it used to be, partly because we have so much more precision with locality information. So this became a really important strategy when I was working on projects with the California Conservation Genomics Project. So I was doing some collecting for Leia glandulosa, Opuntia basilaris, and Iris macrosiphon with my own project and projects with other researchers in the Jepson Herbarium. And so part of this project was we had to go out and find 150 individuals of a given taxon. So for an Opuntia, they tend to stay where you found them. They're long-lived perennials. You can go back year after year and find the same plants. For an annual, like this Leia glandulosa, it could be a lot more challenging, especially because we were collecting during a drought year. And so having to contend with maybe a really narrow window phenologically when we could find plants in bloom, and just knowing where we might find them or when we could catch them at the appropriate stage to make a good voucher specimen and collect tissue was pretty challenging. So we ended up relying on iNaturalist data for this, which was incredibly helpful. And so just kind of some review of the technique I presented in the last segment, going into iNaturalist and subsetting data. We narrow it down by including the taxon of interest and a geographic area of interest. So that brings it down to a more manageable number of observations. So again, going to the filters, downloading this data set, we will, once your download is complete, you can pull it down. And then this time, for the purposes of making a map, I would sort everything by public positional accuracy. So this will be measured in meters. And so this is the handiest thing to do as a first pass, because all of these things with 1 or 2 or 3 or 4 meters of positional accuracy, it's a good indication that that plant is where you expect to find it. Maybe it's 50 meters and you still have a pretty good shot of finding the plant. But once it gets out into 1,000 meters or more, then it becomes a lot less helpful. So this is kind of the first step I would use to sort data like this. And then once I have those data sorted and subset, maybe cleaned up a little bit, I'll go to it's my.maps.google.com and go to create a new map. And you'll go to import and you'll be able to bring it in, I think as a CSV file is ideal. But you could use some different formats. So go ahead and pull that file and upload it. And then the columns to position your place marks are going to be latitude and longitude, or they could be decimal latitude and decimal longitude. So it might select these automatically, or you may have to go and find them, but this is what you'll want to use. And then in terms of titling the markers, you can use anything you want, user login, or sometimes I'll use the positional accuracy to title. This is less important. And then you import all those points and they will appear in the map like this right after you import them. And then you can kind of go through and you can change the base map. So you have a few options there, and you can change the style or the color of the marker. So this is sort of nice. You can, well, this casual ID, those could be in someone's garden. It could have different reasons that they are not research grade. So some of these are research grade, and there's a lot more that need ID. So this is kind of helpful for trying to figure out where you might actually find these plants, or where they might be misidentified, or could be a different species of iris. And then you can also title your map. So once all those are on there, you can click on any one of these points, and then it'll pull down all of these data. And part of that, it'll have a link to the specimen. So you can say, well, this thing seems a little bit out of range. I'm not sure if it's correctly identified. You could actually just pull up the link and then just go online and actually look directly at the observation. If it is misidentified, you could identify it correctly and exclude it from your data set or what you'd like. But it's really nice to have this this spatial organization that links to the specimens just like that. And the map feature on iNaturalist is a little bit clunkier to use in the field. So I like using the Google map for purposes of navigation. And so you can actually just export this map to your phone. And then if you're on a collecting trip, you can plan your route. So, we were trying to find 100 separate populations. So it was really useful to be able to sort of divide this and go from population to population. But you could say, well, you're starting in one place and then ending up somewhere else, and actually, just find that route and enter it into your phone, and it will help you navigate. Or, you could be driving by and looking at the map and say, oh, there was a collection made 1933. And so this isn't just for iNaturalist data. This is for any kind of spatial data with GPS coordinates. So you can use this with herbarium specimens too. And that's where that precisional accuracy becomes really critical, because there is a huge variance on precisional accuracy with herbarium specimens, and the way that they were georeferenced. If somebody tried to say, well, let me just pick the centroid of this place, you can usually go and look at the label, and depending on when the specimen was collected, or sometimes if it's summit of Mount Diablo, it doesn't matter if it was collected in 1895, you have a pretty good idea where the summit of Mount Diablo is going to be. So sometimes a geographic feature will help you even on a really old specimen. Otherwise, you're gonna just want to be careful and thoughtful about that. So being able to sort and filter all that before you make your map is really helpful. So, and then one last thing is, when you have this great map, it's all is well and good while you have cell reception. And then if you lose cell reception, being able to keep those points on that map loaded can fail. So depending on where you're going, sometimes it's just fine and you have good reception. But if you're doing a bunch of botanical collecting in the mountains of California or anywhere else, you might have a harder time. So I like to just take screenshots, just doing the clicking thing with my phone to try to get some points. And so I'll get all those, have them to refer to in case I lose reception and the details of the map fall out. You can still kind of see where these places are and navigate to those places. And truth be told, most plant specimens are collected along the side of the road. So as long as you have the habitat information, a lot of times that roadside locality might be good enough. So that's a short one, and be happy to take any questions about the field press or about map making.